protein. It binds to a protein on the red blood cell surface we call basogen. If you clear this from the parasite will die. And in humans also, if we clear this protein from your system, you will not survive. Because basogen is in every cell. And one of the key proteins is thought to play a role in our visioning as well. So these are some of the things. So one thing people have thought is that if you look at this antigen, it is very small, so it is easy to produce. So here, what we demonstrate is that he is able to produce the antigen very well. Yeah. The problem with we designing vaccine is not sometimes also about you producing the antigen, but in what form it should be. In the normal system, if we do 1:1, we give you the antigen like single one. One thing is that the immune cells will see it, but will it be able to give the enough focusing so that your immune cells will say that, okay, this is something I must invest in and produce enough antibodies or protective mechanism to. So one thing we've realized from the experiments over the years is that if you immunize with just the full-length RH5, you don't have good amount of protection. So the other things we have always been looking at is that, can we then learn from what viruses use? We know the viruses are particles, but then they have proteins on their surface. So if it's a small particle, you can have as many proteins on the surface. So we have this platform we call viral-like particle. So that is what we designed for this. So what we did is that in this case, we use the hepatitis B surface antigen, but then to be able to get the antigen, which is RH5, to the surface, we have to put a tag we call spike tag. This spike tag will allow it to sit on the viral particle surface so that it's easy to study it. So in essence, if you look at the RH5, this is what it is. If you now attach a spike tag, this is the spike tag, this is we have the capture and then the hepatitis B surface antigen in a viral construct like this. So if you put this together, you will now get this. So indirectly, we are mimicking the way viruses are able to expose their antigens, which our immune cells see easily. So this is what we did for RH5. And clearly, one thing is that we could purify our viral-like particle here, but then in constituting it, you will have also your protein in units. One thing we could do is that in immunizing mice, we could show clearly that throughout, if you have, this is your hepatitis B surface antigen here, when you immunize, the focus is not on the hepatitis B. We want it to be on the RH5. So after the third dose, you really get comparable amount of antibody that is produced to the antigen. The other thing is that we could show functionality, that these antibodies are not just produced because anything the immune cell sees, it reacts to. But here, the reaction is very specific and very protective. So if you grow the parasite and you now use these antibodies, you see that there is a dose-dependent ability of the antibodies to block the parasite from entering. And this is very, very interesting and very commendable, because hardly do we achieve this level of activity normally in the lab. So this gave us the tool, and we believe that with this tool, we can now go forward and see how. But one particular thing that has been our individual motivation in RH5 is that regarding you and I, if we sample all of us and we ask the simple question, how many of us have antibodies that sees this RH5? Very, very few of us. But then, if there is any one of us here who sees this protein more, you'll be more protected. So, the simple question is that if we look at the adults, it's easy. But then, when you go to children, the newborn, what will it be? What we have been able to show is that in newborns of parents or mothers who already have malaria, they acquire some level of antibodies to RH5. So the first 6 months level of protection that we always describe, there is a taste of RH5 in the cocktail of antibodies that infant inherits from the mother. So this is one thing that we are now driving down to see how we will be able to design the RH5 vaccine in such a way that it will help not just the normally we look at children under 5, but now also adding the neonates in addition to be able to have very good amount of antibodies in terms of their mothers producing it and then the children inheriting. The other thing we have been doing in addition to the PF-RH5 story is we took advantage of Ghana. There is a unique pattern of malaria. As you move from Accra towards the north, you will realize that for a child in Accra to get malaria, they just need some small amount of parasite, like say 3,000, let's assume. But if you go towards Kintampo, a child will be on the football field playing, but then they have 1 million parasites in them. No sick. Then if you go to the north, which the malaria season is just one, there also you go down a bit. So then looking at these individuals, the simple question is that what allows the child in Kintampo to be able to carry parasite and not fall sick? So we collected samples from Kintampo, collected samples from Accra, and collected samples from Navrongo, and asked the simple question, what is the repertoire of antigens they see? So we got a number of antigens, then we use the criteria of their ability to be used as vaccine candidates. And that allows us to narrow down some of the candidates that I will now be presenting. So here, I'll be presenting one of the candidates we call PF-MAP. What is unique about this PF-MAP is that it is only found in Plasmodium falciparum and Gaboni. So the other parasites, you won't find it inside. But then one thing is that it is also very, very conserved, like the PF-RH5. So what we were able to do is that we could produce the proteins. If you look at the protein structure, it looks very nice. But then the simple question is that, is it stable? That is one of the questions that we've been trying to deal with. The protein has what the central repeat, we call it the armadillo repeats. And one thing with armadillo repeats is that it is a property with proteins that are able to not only be at one location, they are able to shuffle to a number of other locations. So indirectly, it means assuming you are looking at the blast stage parasite, then the forms that the mosquito picks, which we call gametocytes, and then you look at the parasite also in the mosquito. Do we get this protein expressed in that? So what we were able to do is that we could produce monoclonal antibodies here, and we showed that the monoclonal antibodies recognize the antigen nicely. And the other thing is that with the antigen, we could also show here that the antigen is well recognized by the antibodies that you and I acquire just because we live in malaria endemic area. So this is what we succeeded in doing. What we could show is that the PF-MAP antibody recognizes the parasite nicely. And like I mentioned earlier, one property of the armadillo repeat is that it shows different patterns, and that shows that it shuffles from different location to the other. If you look here, you will see something round, round, round. Then when you come here, it's more like dots. It tells you that it moves from being round in nature to being localized to a specific region of the parasite. That is basically what this tells us. The other thing is that if you look at the form of the parasite that is taken by mosquitoes, this antigen is in there. And so far, none of the candidates that we have been using for malaria vaccine exploration have we seen this property. So we think this antigen is unique in its own way, and therefore we will be able to push to see how it will fare in a typical vaccine scenario. We could compare this antigen PF-MAP to already the PF-RH5 that I had mentioned. We see that if you look at, say, among us, many of us will produce antibodies to PF-MAP. So the other thing is the amount of antibodies we produce is more better than RH5. The main thing here is that we could compare the monoclonal antibodies that we produced for PF-MAP, compare it with the monoclonal antibodies available for PF-RH5. And one thing that we can see here is that the green is PF-MAP, and I mean, is it violet or purple? Yes, one of them. Yeah, that is PF-RH5. You will see that the responses are very similar. So with this, what we can confirm from here is that the antibodies are functional. And if you look at, say, the IC50, you will see PF-MAP very, very close to RH5. Therefore, we see it as a promising candidate going forward to pursue. The other thing, just to note, there are 3 parasites that cause malaria dominantly in Ghana. The first, who is the senior prefect, is the falciparum. So anytime you have malaria, the first thing you would think, oh, it should likely be falciparum. You don't know it as falciparum. You say, I have malaria. But likely the biggest suspect is always falciparum. But on the other hand, there are other ones we call Plasmodium malariae and ovale. One thing that is unique about these other species is that this guy, ovale, it is thought that even when you've taken drugs, once they are in your liver, they can decide that, as for me, I'm not going anywhere. I'll just release some few, and the rest will stay. After you've taken the drug and the drug power has waned, then we will come out and see what is happening out there. The other thing with Plasmodium malariae is that it can cause chronic infections, and it has been shown that it is able to drive nephrotic syndrome. And it is able to also cause a number of kidney-related impairment, this chronic infection of P. malariae. But one particular thing we know is that whenever falciparum is there, these guys are quiet. The moment falciparum goes, there is this hypothesis that these guys now begin to come up. So the simple question we're asking here is that with all the interventions, the drugs, everything, focus on falciparum. When we clear falciparum, what will become of these guys? Are we paving way for them to also now assume their senior prefectship? That is the question. So here, what we're doing is that with the currently approved 2 vaccines, the RTSS and then the R21, they target this protein called the CSP. And we know now from the data available that these are able to elicit some level of protection. So, assuming now, if we are to now design vaccine that will target all these 3, how will it be like? So that indirectly you are not just selectively eliminating one, but then you will be driving the whole thing, malaria, not malaria from falciparum down. So here we use the PF-CSP. We also express PM-CSP, PO-CSP. And clearly, if you use any one of us serum, it recognizes them nicely. So we are thinking that if we could design something for PF, we could also do same for PM and PO and see whether synergistically, if we put them together, we will be able to drive the malaria that we want go down to the barest minimum as possible. So clearly here, what we show is that if you look at different people, there are different levels of reactivity that we see to these antigens. And clearly, this is one thing. Franklin, who is a PhD student in the group, is doing. So if you look at it, if you take people from just community, you see some level of antibodies to all these antigens, clearly. Then if you go to the hospital and find people who are sick, you could see that there are different varying levels of responses to this. So clearly what this tells us is that whether we like it or not, we see the falciparum bit. Now also we have shown that we see the malare and ovale bit. Therefore, what this now gives us is that it gives us the ability to now go and look at it in this world. Copy is not theft, isn't it? Sometimes it can be, but in this field of science, it is just good to now use the reasoning to now design similar vaccine construct. The other thing is also we could use the VLP system again and see if we could have a single viralized particle that will have PF, PM, and PO on the surface and be able to see the level of protection this will be able to offer us. So in order not to bore you that much, this works that I've presented has been a long-standing work that has been happening in the malaria lab. So the initial work was started with Dr. Emmanuel Amlabu and myself when we arrived in the country as postdocs at the Awandare lab. Dr. Lucas Amingaitogo is more into the genomics because you need to be able to see the genetic diversities of the candidates that you're working with to be able to see that whether this is more conserved, this is more polymorphic, and therefore which ones you'll be able to prioritize. Abdul is joining the group to look at, when we say vaccine, every one of us have vaccine. We've been told that some have 18 years protection, some have 10 years protection. Are they really protecting us that amount of years? And what shows that that is actually happening in us? So that is his work. Hetty is also looking at invasion-related genes. Yao is the immunologist in the group, and when you talk vaccine, you talk immunology. Joe was a postdoc who joined the group, and he also helped in establishing and leading the protein production platform. Professor Samuel Kusi has always been a long-standing collaborator. We've been working, especially on the epitope discovery. He has been leading this for years, and therefore, we've been really enjoying his wisdom. So you will see that we have, this is just a snapshot of the few that we think, but the samples that we receive from the different hospitals, the different patients, the clinicians, everyone is duly acknowledged, and we thank you.
So I think we're still, as an academic, you went over this time slightly, but I think we are still within time. So we can have one or two questions before we move to the next talk.
Second, just a follow-up question. With the IgG levels that you show in the 3 cohorts, how sure are you that they are sort of like, as I said, again, single infection or mixed infections, and the CSP sort of octology, is there sort of like a cross-reactivity that you might be seeing some signals in the three cohorts?
Yeah. So, malaria, ovale. So one thing that is very clear, and I don't know, but it seems now we are picking a lot of mono infections. The simple question we've been asking is it that we were not looking for them because it is tedious to always look for them, or now we have tools that makes we being able to find them a little easier? So if you go to a typical hospital, now we can see that about 15% of people attending hospital, mostly, will go with co-infection with malaria. Then if you look at the mono-infection of malaria, it's around 3 to 5%. So that is what we see. Ovale is around 3 to 5% in there. And we ever had a case of a person who had all the three in him. And it was a child. So we are seeing them more now. We don't know whether they are coming up or now the tools that we have is allowing us to pick them up easily. In terms of the antibody, one thing is that the PF-CSP has the NAND repeats. One thing is that if you look at the repeat regions of PM, PO, the signatures are different. So if you do a structural modeling and you want to see the level of alignment between these three, it is very weak. And also, even if you look at amino acid alignment, it's around 30 to 40%. So one thing we have been doing is now looking at cross-reactivity. So one thing we believe is that there is not so much of cross-reactivity, but then the other thing is the level of cross-reactivity based on amino acid or it's conformational epitope based. That is the other thing Franklin is now driving home on. And we believe by the end of the year, maybe he should have a few papers out on this.
Okay, I think Nana Prof has a question, and then after that, Fahan. Mike.
So when is your candidate going to get clinical trials?
No, so one thing is we now have the construct, the VLP constructs. So we are, first of all, you have to look at the construct that you have. No, the construct, how much antibodies it will elicit. So that is the first thing that we want to do. Okay, so for PF-MAP and PF-RH5, if we have the money, yes, if we have the money, in 3 years, we could begin to test it. Yeah, because as a start, I don't have the money.
Okay, and I think Fahan had a question also.
Okay.
Okay, the question is like, when we talk about malaria, and we are talking about a lot of discussion is happening in malaria, the problem was like tissue-sensitive immunity was always a problem because generalized immunity is easy to achieve and tissue-sensitive immunity is always difficult to achieve. Yeah. Now you have achieved it. And another problem is like, I will echo Professor Ampofol, so that first of all is like lab, when you are developing a titer in lab and shaking flask, it is very easy and it goes very cool. But when it goes to the scale up, yeah, the problem which comes in it that how you control the contamination and how you pure that characterization of that product is very well done unless it reaches to the clinical trial level. So no doubt you do not have resources right now. So have you tried on a liter basis that, okay, the scale up has gone perfect and you are very confident that when you have the funds available that you can go for the clinical trial?
For the VLP constitution as a vehicle, yes, that is very, so I mean, just to piggy on what you mentioned, I think that is why we are solidly happy for the initiative of the National Vaccine Institute to have a pilot production lab. So like this, we can easily go scale it up, say to, now we work within 10 liter range. If we have, say, 50 liters, how much will we be able to get? And that will be a very good thing to do. So we're still open. And like you will see in my acknowledgement, we're still working with Oxford, the Jenner Institute, a group there which is helping us to see how much we will be able to scale up.
Then also, sorry, doctor, there's another a discussion which is going on right now is antibody vaccine conjugates. Because vaccine is available, R21, it's showing good results. Then antibody you are developing. So are you also working on the antibody vaccine conjugate? Because that is the future, which is basically right now predicted in the next 10 years.
Yeah. I mean,
so have you started work, and what is your work right now there?
So you will see the approach we use to many of the antigens that we're looking at is we don't just look at the antigen, we try to also look at producing monoclonal antibodies to it. The specificity of the monoclonal antibodies is something, and the functionality, that is where we're looking at. So once we are able to have a very, very functional monoclonal antibodies, then you can now look at the antigen antibody conjugate and then be able, and it is actually, there are new technologies that are also now evolving on how to really have this constituted and being stable also.
So that's what I was kind of alluding to. You guys do a lot of basic science stuff. You showed us the epitopes, you showed us the immunodominant regions, etc. We want to push you now to have a targeted, kind of pipeline, whether it's the antigens that could be candidate, vaccines, or whether it could be monoclonals that could also be used for therapy. So we want to move away from the traditional basic science, or you can continue the traditional basic science, but we want it to be focused so that then we can then put in resources that will enable us to say that, okay, after 2 years of supporting basic science research, we now have a pipeline of particular products which we can move forward, from discovery actually now to commercial, feasibility or viability. That would be very important to have the resources allocated to support you. So we need you to develop those plans in addition to looking at exciting or innovative basic science that, answers a lot of the questions, but also looks forward to the end product. That is very critical, for us to go forward. Thank you.
Yeah, thank you.
All right. Thank you, Dr. Aniwe. So in designing the schedule and the speakers, we also wanted to talk about one of the reasons why we are here today, as our guest of honor intimated before, is that of the COVID-19 pandemic, which brought a whole host of issues regarding vaccination, and also coincided with a lot of mis and disinformation. So one thing that we are also doing along the lines here is a project that ultimately leads to the production of monoclonal antibodies from patients. So Dr. Irene Owusu is going to start us off from the beginning and head towards there. So I'd like to call her to come and give her presentation.
All right. Good morning, everyone. I am Irene Owusu Amwako, and I'm a virologist, but here at WACB, I work as a postdoctoral fellow. And today, I'll be sharing with you work that is ongoing on the expression and durability of multivariant SARS-CoV-2 neutralizing antibodies after people have been exposed to the vaccine. And then we'll have some cases as well. This doesn't work. So last year, sometime in March, WHO declared the end of the COVID-19 pandemic. And, excuse me, sorry, this is just not working. So we were excited to hear that the WHO had ended or declared the end of the pandemic. But then it's also important for us to keep in mind that the end of the pandemic does not mean that the virus has been eliminated or eradicated from the system, meaning that as long as there are people who are susceptible to infection and then the virus is still in circulation, the severe effects or the risk of getting COVID-19 and then certain outcomes is still possible. Now, we all agree that during the pandemic, the introduction and administration of the vaccine played an important role in helping to reduce the severity of the infection. But so far, we don't know for how long this protection from the vaccine and even exposure to SARS-CoV-2 would protect people. And so it's important for us to know what the durability and then the breadth of vaccine immune response after exposure to the antigen is. That's why our projects worked on. We try to look at neutralizing antibodies that people who have been exposed to SARS-CoV-2 antigen would get, and for how long the neutralizing antibodies would stay in these people to protect them. This information is always important to also inform vaccine schedules and how much antigen or vaccine doses should be given. So for a vaccination to be successful, we all know that among other things, two main things are what we'd look at. There should be memory cells produced, or the vaccine should be able to induce the recipient to produce memory cells, and also the antibodies produced should be able to neutralize the pathogen. Now, these neutralizing antibodies are able to, So these neutralizing antibodies are able to reduce or prevent viral infection by various mechanisms. They can either prevent the attachment of the virus to the cells for entry, or once the virus has attached, they're able to prevent entry or penetration of the virus. And in other instances, the neutralizing antibodies interfere with the uncoating of the virus for the replication to occur. Now, in all these things, neutralizing antibodies always recognize the epitopes of glycoproteins or surface proteins of the virus to be able to interact and then inhibit their action. So our main project was to first identify plasma from patients who had been vaccinated or had the infection, and identify certain plasma samples that could be able to neutralize different variants of SARS-CoV-2. And to do this, we used the live virus neutralizing assay. Here at WACBIB in our lab, we have various systems for testing for neutralizing antibodies. We have the pseudovirus system and the live virus system, but data I'll be showing you today are data from the live virus system. So for the live virus system, once we get samples, we serially dilute the plasma samples, then incubate them with a known amount of virus, incubate for an hour before we put them on viro cells. We use a viro cell system, and we do the infection for up to 3 days, and after that we stain and look for where there's virus infection or not. So where there is no virus infection, like the deep blue wells, that is where we know that there are antibodies or neutralizing capacity of these plasma samples that prevent the virus from infecting the cells. And then we quantify and then get the neutralizing titer. So it's a straightforward method. Compared to the pseudoviral system, this live viral system is more cheaper, and it's easier for us to do here in terms of logistics and then costs. So for this part where we wanted to identify plasma samples that have neutralizing antibodies, we collected samples from patients who had COVID-19. So, and we sampled across the different waves of SARS-CoV-2 from wave 1 through to wave 5. Now, almost 200 samples have been tested so far, but then in our biobank, we have thousands of these plasma samples that we are yet to test. And of these, majority of them were unvaccinated, while a few vaccinated. And that is how I'll be analyzing the data to try and make meaning of. So speaking of the waves, I grouped the samples in waves based on how the times that they were collected, okay? And so for the periods here at work, we were able to find that during wave 1, that span between January 2020 and November 2020, the dominant variant was B1.1, and then the peak period was in July 2020, and it goes for the others. So you find that wave 2, the dominant variant was Alpha, and then wave 3 was Delta, then 4 and 5 were Omicron, but different lineages of Omicron. So this is how I grouped my samples according to the times that they were collected. And now for this project, I've tested so far just 4 variants. So the wild type that has the D614G mutation, and then we have Beta, Delta, and Omicron. Now you realize I put Beta in color because we did not record Beta in Ghana, but then I'm testing for Beta or neutralizing antibodies against Beta anyway. And looking at certain spike amino acid substitutions, as I mentioned, the neutralizing antibodies would normally interact with surface proteins. And in the case of SARS-CoV-2, it's the spike protein that is one of the important proteins for immunogenicity. And so looking at the spike protein of these 3 variants that are used, how they differ from the wild type, you find that the Beta has just 15 substitutions, while the Omicron has 58. So we see, which is more logical, that as the new mutants or new variants come up, they have more substitutions or more mutations from the original. And so in this case, Beta will be more closely, will look more like the wild type. It's closely to the wild type than Omicron that came later. And so this is something we'd also have to keep in mind as I show the data. And so when we ran the live virus neutralizing assays for the different samples against the different SARS-CoV-2 variants, I found that most of the donors that we had were able to produce neutralizing antibodies against these 4 variants. So we were able to identify plasma samples that had cross-variant neutralization. And across also the different waves. So you see that as the waves increase, there will be an increase in level of neutralizing titers, which is, it makes sense because as the waves increase, people are exposed to more of the variants, and so there's continuous antigen stimulation in logic. But then one thing I realized that struck me and then that we want to look into is for wave 2, we find that, so logically we would expect that from wave 1 to wave 2 there will be an increase. Then from wave 2 to wave 3 there will be an increase, but it doesn't happen like that. There is a dip in wave 2 and before the increase starts again. So you realize that people who were exposed to the variants in wave 2 might not have better neutralizing capacity as people who are exposed to the other waves, or to the variants circulating in the other waves, okay? And so the question is it true that variants in wave 2 are likely to elicit poor neutralizing antibody capacity? Now, to do this, we would want to even look into testing neutralizing antibody levels against the variants that were circulating during the wave 2 pandemic wave. And these variants were alpha, eta, and then B1.318. These were the most dominant variants circulating in there. So my question, and for my next step, is to look for whether these other antigens or even these virus variants are able to elicit enough neutralizing antibody capacity. Okay. So knowing that we have patients who are able to produce neutralizing antibodies against different variants of SARS-CoV-2, the question was whether vaccination will play a role. So now, before I showed you samples from all the waves put together, there were a mixture of people who were vaccinated and unvaccinated. So I decided to separate that and then see what that would look like.